So we can move on to part 2. So this is going to be explaining how to process FastQ files, actually starting from FAST5 files, to get to an assembly for use in downstream applications. So I've put together some test data. It's tuberculosis data. For the purposes of training, it's just one file because if I did any more than that, it would take forever, and we don't want to be sitting here forever. It's also important to remember and note down which sequencing kits that you used. So for instance, in this, I use the rapid barcoding kit, and that's the number of it. And I also use the 9.4.1 flow cell, and it's important to know the type of flow cell as well, because it changes how it processes the data. And as I mentioned before, the programs used are not necessarily the only options, and the code that I have used is not necessarily the only way to do it, but these are methods that I've used and they work, and so that's why I put them. Some programs use Windows and some use Linux and Windows. Any of the code I've color coded, so just to sort of explain different examples, I've used a Windows command for Guppy, and that's in yellow, and any Linux commands are in grey. Hopefully that will make more sense as we go through. So just wanted to introduce you to Guppy. As I say, it's an Oxford Nanopore program, and it has several different features. And I already mentioned it's integrated within Minnow, so it already kind of runs or sequencing. You might find that the basically powerful computer, or you have GPUs, it can be quite slow. So you can stop it and start it individually again. And that can speed it up a little bit. So you need at least 8 gigs of RAM, but Guppy does run on Windows, Mac, or Linux, so you have different options. And as I mentioned already, Guppy has different functions. So it has the basecaller, which converts the FAST5 files into FastQ files. It has barcoder, which demultiplexes, so that separates your barcodes into different folders or directories. It also trims as well, so it trims off the barcodes. And then there's also the aligner, so Guppy can align to a reference genome, but I think most people still use Minimap2 at the moment, because I don't think the aligner has been particularly widely tested yet. But I haven't used it yet anyway. So to install Guppy, if you go to the Nanopore community website, and I thoroughly recommend having an account on the Nanopore community website anyway, you can download it straight from the website. Or you can also install it directly using the command window. And if you go to Guppy Nanopore documentation, you'll find that there's a page for Guppy, and it explains all about how to install it and how it works. And I think there should be So here I've put kind of the basics. This is the Windows version. So to begin with, you've got the location of Guppy basecaller. So Guppy basecaller, or Guppy _ basecaller, is the program that you're wanting to use. If you haven't sort of linked it, then you need to put the whole of the pathway in, so C drive /document/ wherever it is. Or if you have linked it, then you can just put Guppy _ barcaller. And I think because it's Windows, you need to put .exe to point out that it's a program. Num _ callers is the number of callers or threads. So this depends on how powerful your computer is, and I've put more information in the PDF, but basically that's how much of the memory is used simultaneously to do. So we've got 10 files. Each of those 10 files will be done one after the other. But if you up that to 2, then it will do 2 at once, 2 at once, 2 at once. And if you obviously up it to 10, then it will do all 10 in theory simultaneously. But it's related to how many cores your computer has. So mine is a Core 7. So I think in theory I could have up to 7. It just basically means if you have a more powerful computer, you can do things simultaneously. And obviously there's an awful lot of data going through these programs, so it's quite useful to speed that up. So the -I, sorry, is the input folder and address. So you want your FAST5 files. Often if you have cancelled calling from Minnow, then it will create a file called fast5 skip. But wherever the folder is for your fast5 files, then you just need to add it here. And then S is the output folder, so that's where you want your fastq files to go to. And then C is the configuration file. So you can see here the 9.4.1, that's the the flow cell that you, or the type of flow cell that you want to make sure that you get it right, because there's slightly different chemistries. Fast denotes the type of base calling that you want to do. So the two options are fast or high accuracy. Obviously fast does it quicker, but high accuracy is slower, but in theory more accurate. And then this relates to the type of kit that you used. And again, if you look on the Guppy documentation on the nanopore community, it will explain the different sort of numbers and the different configuration files. So now I'm going to try, and let's hope this actually works. So this is the, this is kind of a, here's one I made, this one I made earlier. So you can see that this here is the Guppy base caller pathway, because there's a gap here between program and files, because obviously that is a general file and I don't want to change it. For instance, if you put quotes around it, then it treats that as a single command. If you didn't put quotes around it, it would see C program as one and then files, Oxford Nanopore as a separate one. So it would count them as different things and it wouldn't work. And then you'll see that my input folder is my tutorial test and my fast5, and then my fastq, and that's my configuration file. So I'm going to copy and paste that, and in theory, this should hopefully work. So this is my command prompt. So this is my Windows version. And if I just bring up this file here, so you'll see this is my fast5 file. So this is the input folder. And you can see here I've got one fast5 file. And this is my fastq folder. So this is my output folder, and this is where I want it to go. And so if I paste that into here, please. No, okay, try again. If I paste that, yeah, come on, there we go, and press enter, hooray! So you can see that that is now base calling. Because it's only one file, it shouldn't take very long, but you can see it adds information files first. And once it's got a bit further, you should see that the fastq file will appear. So whilst this is doing it, does anybody have any questions so far? You can also do this with Linux or Mac. So it's the same principle. You might just find that the input and output file directory addresses are slightly different.
Hi, Lindsay, can you hear me now? It's Zara.
Yeah, I can, yep.
Okay, well,
Sorry.
It's very good and really clear and no questions so far.
Fabulous. Nobody was saying anything, so I was a bit concerned that I was just waffling to myself. But that's good. So you can actually see it's telling you information about your file anyway. So records per file, 4,000. That's the default amount of reads that I mentioned before that Minnow puts into each different fast5 file. So you've got 4,000 reads within each fast5 or fastq file. So you'll probably get hundreds of FastQ files from each barcode. But remember that within those, you will then have 4,000 reads. So you've got basically whatever it is, 100 times 4,000 or 200 times 4,000. So it's quite a lot of data that you're getting. And depending on what type of sequencing kit you use, so if you're using a PCR-based kit, you will get probably a fairly uniform amount or size of reads. So for instance, if you use the RPB 004 and use the long amp, we've been getting somewhere between about 3,000 and 5,000 KB as our read length. And so all of those will be roughly the same size. But if you're sequencing directly from native DNA, so stuff that you just directly extracted or without sort of specifying size, then you can get all sorts of lengths of DNA. So for instance, some people can get up to sort of hundreds of thousands of bases. Some of them will be a lot smaller.
Lindsay,
Great, so that's...
Lindsay?
Yes.
It's
yes.
back up. Can you hear me?
I can hear you.
Okay, it's back up. Okay, I have one question about the trends per color. Here.
What, sorry?
The trends per color. In your screen, I find it's 4.
Threads per caller, yes, sorry, yeah.
Okay, and you say it depends on the power of the computer. If I have computer of 8 gig of RAM, how many color I can put?
So it's not to do with the RAM, it's to do with the cores. It's in the PDF, but your computer, it might have a sticker on your laptop on the front that says, so mine says Intel Core i7. And also you can identify it on the settings in the computer, but I believe that mine could in theory do 7 parallel bits, things at once. So the smaller or the less powerful your computer is, the less cores you are likely to have. If you have an enormous gaming computer, then you're likely to have more, or you're likely to have a GPU, a graphics processing unit, rather than a central processing unit, and it will be much, much faster and more, everything will happen at once more.
Okay.
Does that make sense?
Yes. If I'm following very well, it's not depend on the RAM, but in the core. And I think it's right on the computer, Intel Core. Okay, that's right. Now I know. Okay,
generally, the more RAM you have, probably the more cores you have, because that generally the bigger the computer, the more powerful it is in every aspect. But as I say, on the PDF, I've specifically put how to check how many cores you have. So you can have a look on there, and also we can chat about it when we're next in.
Okay, that's fine.
Does anybody have any other questions? Well, so it seems to have finished. Excellent. So now you can see in our output file or directory, we've got pass and fail, and I'm hoping that because we've only got one, there it is, it will be in my pass folder. So that now is the FastQ version of the fast5 file that I showed you earlier. So what Guppy has just done is change those squiggles into A's, T's, G's, and C's, and that's what we'll find in here. So if I open it with, not Microbe, Notepad, there we go. So this is all of my sequencing data, and you can see it here. We've got, and in theory, this should be 4,000 different reads. I won't bother to go through them all, but that's basically what you'll see. Great. So let's go back to this. This is, as I say, this is Windows version, and this is how Windows likes to have it written out, the file addresses. I know that Linux does it slightly differently, but I will show you a version of the Linux kind of command line as we go through. So just to explain or show the differences, if you copy and paste directly from Windows, that will work within the Windows command line. So, sorry. You can also, as I say, trim. So what you would do first, if you had lots of them and they were all within a same folder and you didn't know which barcode was which, you could also use Guppy barcoder to demultiplex, which means to separate them into the folders. I only have one, so I don't need to do that. But what I will need to do is trim my DNA to get rid of the barcodes. So the barcodes are basically like primers, and you want to get rid of them because they're not part of your actual genome. So again, just to explain, going through the kind of basic code, you've got your address of where the Guppy barcoder is stored. You've got the address of the input file, or the input folder rather. And actually that's wrong. That should be the FastQ output from basecalling. And then the address of the output file. So that's where you want to put it. So R is something we can introduce here. R means so if you put R, that means basically the folder. So that's really useful. For instance, as I said, if you've got a folder that says barcodes, and then within that folder, you've got all of your individual barcodes, so barcode 1, 2, 3, etc. You can just put in as your input address barcodes, and then it will go through all of the individual folders within it, and it will do, it will trim everything that it finds within it. So that's a really useful command. Then you get the configuration file, and you can check on the Guppy page which ones you need, but I think for trimming it's just configuration. And then you need to tell Guppy barcoder that you want to trim barcodes, and you also need to tell it, sorry, there we go, configuration file, we need to tell it to trim, and we also need to tell it what type of barcodes or what kit it was that you used. So as I mentioned, I use the Rapid Barcoding Kit 004. If you don't tell it which kit to use, and this is more important when you demultiplex, if you don't tell it which kit, it will automatically search for 96 different barcodes, because there is an extension pack that has 96 different barcodes. So if you don't tell it exactly which kit to use, it just goes through all of your data and checks for barcodes. And obviously it's inevitably going to find those small bits of code within your actual genome. So if you don't tell it which kit you use, it will output to 96 different files. So I thoroughly recommend not doing that. If you tell it which kit to use, it will output to the actual number of barcodes that you used. If you use 96, then that's fine. But so it's just useful to tell it, give it as much information as possible to give it the chance to do what you actually need it to do. I won't show you how to trim because it's very much the same as doing the barcoding. And also it won't be a very interesting file. It will just be the same FastQ file, but without the bits on the end that it's trimmed off. So we can move on to QCing. There are different programs again for QCing, but one option is FastQC. Which is a simple way to do quality control checks on raw sequence, to be aimed mainly at Illumina data. So you have to kind of, and also your input data might not necessarily be, you have to sort of understand what you're putting in to begin with, so that you understand what the sort of results are. As I say, if you're doing a PCR based kit, your DNA is all going to be roughly the same size, about 3 kb. If you're doing native DNA, you might find you've got some that are only a few hundred and some that are hundreds of thousands. So you just have to be aware of what you put in when you look through the quality check output. So requirements that you need to have Java installed. You need to have at least 8 gigs of RAM, which is most normal laptops. And you can use this using Linux, Mac, or Windows. To install FastQC, you can go to the FastQC website. And again, I think there should be a link in the PDF, and download the zip file. You can then navigate to that folder and run install. So this is on Linux now, and this is how you install it. And if you get an error, it might be that you need to update whichever Linux distribution that you're using, say Ubuntu, which is what I use, and you just need to update. And I believe you need to be the administrator or have administrators to use. So this is how FastQC basic code works. This is the name of the program. So you can either, as I say, link the path, or you can navigate into the folder where the program has been stored. This is the file path to the FastQs that you want to QC. And you'll see now that this looks slightly different to the Windows one. It's slightly complicated by the fact that I have a Windows computer and I'm using the subsystem for Linux. But the way this works is you have to change around all of the slashes and add mount to begin with, which means that your Linux sort of interface is connected to your actual computer system. So this now then will be my C drive, and I'll find where my FastQ files are. And again, so yes, if you put a star, that means that you want it to do whatever it is you're asking it to do to all of the files within that folder or directory that have the extension FastQ. And these are the number of threads. So again, you can change that depending on how powerful your computer is. So I can do this now. Where's that gone? There we go. So I can navigate to my file. So CD means navigate to. So I can now, that's not what I want Ubuntu. So this is my Ubuntu setup. And you can see that just by copying it in, it's now moved me into the folder that has FastQC in it. I can then copy and paste this in. Where's it gone? Let's open. I have navigated into the folder where FastQC is. So you can ignore all of the, the rubbish there. And now in theory, there we go. Basically the problem was I was stupid and I forgot to trim the file. And because I didn't do it, there wasn't a FastQ file in there. But now, because I've moved it, there was a FastQ file and it was able to do something. So there you go. There's a good problem solving exercise. So now you can see that it's created this FastQC HTML folder and file. And so if I click on that, it will bring up this FastQC report. As I say, it often says Sanger and Illumina, and I'm not sure whether there's anything we can do to sort of change that. I did check, but the person who I asked all my questions about this to thought that you just did it and hoped that it would work all right. So the number of sequences we've got within that file, 3,791, so nearly 4,000. So this might have been the last FastQ file, perhaps. None of them have been flagged as poor quality, which is nice. And you can see here the sequence is somewhere between 36 and 15,000, and that's PCR, and therefore we've got varying ranges of size here. And the reason that a lot of these will be marked as red or failed is because it thinks it's Illumina data, and so it's confused by the fact that we've got some really weird sizes. So it's important to sort of consider what you've got, as I say, before you go into it. And this is TB, so it's got quite a high GC content. So you can go through, sorry. This just shows you the quality scores for the different positions within the sequences. So there's a really good tutorial. So Sylvia put it onto the PDF. So if you want to go through in more detail about what these show, then I recommend looking through that. But you can see here it's got the mean quality score, basis per sequence, and you can see that generally it's quite even. Obviously there's more G's and C's, but at the beginning, the first few ones, it's a bit all over the place, but usually at the beginning and the end of sequencing, it's a bit dodgy because it starts to identify what it is, but generally that's fairly uniform. GC content, and then ends. So if it doesn't understand what the base is, I think this is more for Illumina because I'm not sure that Nanopore does this, but it's got no ends where it doesn't understand which base it is. And here you can see the distribution length. So as I mentioned because it's native DNA, so we've got lots that are really small, but actually we've still got some that are going well up to sort of the 15,000 base pairs. And if you had, as I said, PCR base 1 and your bases, your lengths were roughly about, say, 4,000, you would get more of a normal sort of bell curve at around 4,000. So that sort of entirely depends on what it is that you've sequenced. And then, yes, there's various different bits of information. This sequencing adapter content, these are all Illumina based again, so you should hopefully find that you don't have any if you're doing nanopore, but if you're doing Illumina, then you'll find presumably that you'll have some if you haven't trimmed them off. So that's FastQC report. Let me go back to here. Then you've got MultiQC. So FastQC looks at individual FastQ files and gives you results for individual files. You can run more than one together, but it gives you information for individual files. MultiQC searches a folder for lots of different FastQC reports and compiles one big report. And again, you need Java, 8 gigs of RAM, and you can use any of these operating systems. If you want to install it, you can use Conda using this code here. And then you can navigate to the folder where where your files are. MultiQC is the name of the program, and then dot means do MultiQC to all of the files in the directory that you're in. So I won't do example code because I've only got one, and I also have here's one I made earlier to show you. So, but if I wanted to do this, I would change directory into the folder that I was looking for. So if I had multiple files within my trimmed folder, I would move into that folder, and then I would run MultiQC and the dot. But as I say, I have, here's one I made earlier. So it will output this HTML report folder file, and this is what will come up. So you can see that these are the different FastQ files that I have done from, this is from a different project. It's showing you the different lengths of each of the different average lengths of each of the files, different GC content, and you can edit this table and show different sort of columns. And it also shows you the number of reads within each of those files, gives you quality scores. It's very similar to the FastQC, but it does it for multiple files and provides sort of one report for multiple files. So that's Fast and MultiQC.